Oh,
All right. So thank you everyone for joining us. My name is Mark Howard. I'm the Executive Director of the Candlewood Lake Authority and I'm hosting this webinar. So to make the presentation of the webinar go smoothly as possible, everybody is muted and video has been turned off for guests, but we do welcome and encourage your questions. So if you could type them into the chat, and then at the end, Neil and Steve will answer the questions for everyone here. So we'll answer them all now. This webinar should last most an hour, including the questions. And I just want to take a second and introduce you to our two presenters, Neil and Steve. Neil Stalter is the Director of Ecology and Environmental Education for the Candlewood Lake Authority. His background is in hydrology and environmental science and policy, with a focus on water resources. He earned his bachelor degree in ecology from the University of Rochester and his master's from Columbia University. Neil grew up on Canisius Lake in the Finger Lakes region of New York and brings substantial scientific expertise and experience from his educational and professional background. Steve Slugy is a New Milford delegate for the Candlewood Lake Authority. He is also our Watershed Management Committee Chair. Steve's background is in geosciences, and he's taught in the Bedford, New York schools on the Juneau ice fields in Alaska, and in a Cornell University program on the Hawaiian Islands. He is currently a National Geographic expedition expert in the National Parks out west, which is I hope I get to do something around that. Steve's family has been enjoying the lake since the mid-1930s, and he's third generation of his family to occupy the lakes, the family's lakeside home in the lake. Steve and Neil have been spending lots of time out on the lake together, gathering water quality data and monitoring invasive plants and animals in an effort to understand and recommend the best management practices to various stakeholders of the Candlewood Lake. So with that, I will turn it over to Steve.
thank you, Mark. That was a very nice introduction. So thanks everyone for joining us tonight. So this is our environmental report card and check-in webinar. And basically what we're going to be going over today is a few things, but we'll be focusing a lot on our normal water quality monitoring program, sort of explaining what it is that we're looking at and keeping track of. And then we'll show you some results from last year, some preliminary results from 2021 this year, and talk about a few other key environmental aspects like invasive species and blue-green algae. So thank you all for joining Steve and I. And just these are the things I mentioned, and this is sort of the agenda in order. And so we'll give you an idea of what our monitoring program is, followed by telling you exactly what a healthy lake is, sort of defining what that means. So you have some context for the results that we'll be showing you from last year and this year. And then we'll cover those 2 years. And then finally, we'll end off with an invasive species check-in. I know a lot of you are probably wondering about zebra mussels as well as other invasive species, so we'll talk about those. And blue-green algae, current status, and some information about that. So looking forward to it, and we'll just jump right in. So to start us off, I wanted to give an overview, a general overview of what it is that we're doing out there to monitor the health of Candlewood Lake. And so if you look at the map on the right, you'll see the 5 locations in red that we sample twice per month from May through October. And we'll go into a little more depth than exactly what we're sampling, but you can see there's the 4 locations on Candlewood, the 1 location on Squan's Pond here. And then, I should mention that historically, this has been done since 1985. But 2021 is the first year where we have had the time and the manpower, thanks to Steve's assistance to me, to do a second sampling. So we've basically doubled our monitoring, and this is the first year that we're doing it. So that's very exciting. And then this purple dot here, this CT, that's the Candlewood Trails location. We use that for some experimental sampling projects, sort of just exploratory, get an idea of some really cool stuff, and Steve is going to share some of that with you later. So we'll move on to talking exactly what it is that we are monitoring at each of those locations, and I'll hand it over to Steve.
So on that bi-monthly sampling, or monitoring, what we do is we drop a probe. You'll see it on the picture on the left there. We drop a probe down into the water and stop every meter on the way down. And we take the lake's temperature. We measure the oxygen levels in the lake. We measure the pH, the acidity or lack of acidity in the water. We measure the conductivity of the water, and that's saltiness. And you're going to hear us talk about saltiness tonight. And it's not saltiness that you can taste. It's not like as salty as seawater, but all fresh water in streams always has some degree of salt in it. And we are watching the saltiness of Candlewood carefully because it's one of the things that's changing. We'll also look for chlorophyll A, and we also look for chlorophyll A, which is basically how much algae is there. And we measure the blue-green algae as well. The little diagram in the middle, that's me in the back of the boat and Neil in the middle. Neil's dropping the probe and I'm collecting some water samples. The cool thing about that probe is that we connect it to an app on our phones, and we can gather the data that way and then upload it to a spreadsheet or whatever afterwards. But it's nice because we can look at what we're the data that we're collecting in real time, and that helps us see what's going on in real time. So it's an interesting way to do things.
Awesome. And then the other portion that we're doing at each of those locations is we are taking physical water samples, and we take those to a lab in New Milford to get analyzed for a number of metrics. We analyze chlorophyll A through the lab samples as well. We have a new probe, or a new sensor, I should say, on our probe that measures chlorophyll A, and we're working on figuring out exactly the best way to compare the lab samples that we get with our new sensor. So that's still under process. So we're doing both for the time being. We also take sodium, calcium chloride, and magnesium ions. And so those are basically charged ions in the lake that have important properties for various reasons. Different ions have different properties, but one of the main things is Steve brought up the saltiness. These are many of the ions that are contributing to that saltiness. So, sodium chloride, that's your classic table salt, and then calcium and magnesium are also salts, but they play a little bit of a different role. So we want to know exactly which of these ions are sort of contributing to that saltiness the most. And then the other final two are nitrogen and phosphorus, and those are two critical nutrients that plants use to grow. And in the same way that plants use them to grow, so does algae. So nitrogen and phosphorus are natural in lakes, but only to a certain level. And humans are sort of notorious for adding nitrogen and phosphorus to lakes very quickly. So we want to keep track of exactly what's happening with those nutrients. And we'll talk a little bit more about those and their impacts a little later, but those are highlights. You can see the tool that we use over to the right here. This is called a Van Dorn bottle. It's sort of a nifty little gizmo. You open up the two end caps here. You can see the blue end cap, and you open it up, and then you drop it down to the depth you want to take the water sample from, and you drop a weight down the rope, and it closes that end cap. So then you have a water sample from that discrete depth. So this tool allows us to take water from specific depths that we're interested in looking at. And so we take water samples from 3 depths, one near the surface, 1 meter depth, one at the middle level of the lake, which changes based on the temperature of the water as it goes down with depth, and then one at the very bottom near where the water ends and the soil begins. So we have a total of 15 samples generally that we take to the lab every sampling event to get analyzed. And so I mentioned the nitrogen and phosphorus. So when we're talking about a report card, obviously we need to figure out some way to actually grade our lakes. And so one of the easiest and most informative ways to do that is through trophic status. I know these seem like big words, but all lakes have sort of a natural life cycle. And a lake begins as this oligotrophic lake, which you can see here is sort of this beautiful, deep, clear blue water. And think Lake Tahoe with a oligotrophic lake. And then over time, naturally, lakes actually begin to fill in with plants, with sediments, with algae. And then eventually, and we're talking on a time scale of tens of thousands of years, even hundreds of thousands of years, depending on how large the lake is. Eventually, it will become eutrophic, which is sort of a little much shallower, swampy, very filled in. So, after you're talking about 100,000 years, this beautiful oligotrophic lake will naturally sort of become this eutrophic lake. The reason that we look at this so carefully is human impacts. So development around lakes, all sorts of pollution, anything like that can actually speed up this process. So instead of talking about hundreds of thousands of years, depending on the lake, it could change to hundreds of years or just 1,000 years, depending on the size and how often it's used and how much development there is. And so we can sort of grade any lake, including Candlewood, on this timeline between oligotrophic and eutrophic. As sort of a shorthand, we can say that an oligotrophic lake is very healthy. It's, easy to use. It has a lot of ecosystem biodiversity. And then slowly, as it gets older, it may become less healthy. So we're trying to slow that process down instead of speed it up. And so you can see the metrics that we use to sort of grade. There's the phosphorus and nitrogen that I mentioned. There's the chlorophyll A, which is the algae material. And then Secchi disk transparency, which is a tool that we use to measure how clear the water is. So as phosphorus and nitrogen go up, you're going towards eutrophic. As chlorophyll A goes up, you're going towards eutrophic. And as transparency, as water clarity goes down, you're going towards eutrophic. So this is how we're going to sort of grade Candlewood for 2020 and 2021 so far. And so I know I'm doing a lot of talking, but I made all these graphs, so it's easier for me to go through them. But this is one more graph that I think is really informative and gives a really good idea of sort of how a year can compare to other years on Candlewood Lake. And so on the x-axis here, you have chlorophyll A, which is algae material. And as you go to the right, you're getting greener water, you're getting more algae. And on the y-axis here, this is Secchi depth. So as you actually go down this axis, the water gets clearer. So think of, the top of this graph as being the top of the water. As you go down, the water is going to be clearer. So down in this corner, this is clear water with very little algae. And over here on this side of the graph, this is green water with a lot of algae material in it. And so we can use this to sort of get an idea historically how each year sort of fits into the larger picture of Candlewood Lake's water quality. And you can see this is 2020. We'll show the graph a little bit later and 2019. But you can see 2020 actually was an excellent year as far as water clarity, chlorophyll A. We sort of had a little bit of a banner year. We set a record for the highest water clarity measurement we've ever taken since monitoring began last year. So very clear water year in 2020.
And Neil, I think what's important to point out in this is that from one year to the next, the water quality can vary tremendously. We can have a kind of murky year, and then the next year can be simply awesome. And one of the reasons we're doing all this monitoring and kind of looking at weather and everything else, we're trying to figure out, maybe not to the point where we can predict, but maybe where we can have some kind of impact on all the factors that will either help algae grow or slow it down.
Right. Yeah, you'll notice that this line is not a trend line. This is just showing the relationship between the variables. You'll see here by 2020, you've got 1989. And then, up here in the green water, more algae, you have 2013 and 1986. So you're not talking about a trend with patterns here. This is sort of just putting in context how clear and recreationally nice the water is from year to year. Oh, and this one's me too. And so this is some good news, actually, this graph. So we wanted to illustrate the phosphorus, that key nutrient that I mentioned, as an important metric and what's been happening since monitoring began in 1985. And the way that phosphorus gets into the lake, generally, it can enter naturally through the soil, but humans often sort of accelerate the inputs of phosphorus into the lake through fertilizing their gardens, fertilizing their yards, and then when it rains, the rainwater picks up that fertilizer, which contains or probably contains phosphorus, and it brings it directly into the lake. Because the same phosphorus that the terrestrial plants love, the aquatic plants and algae also love. And so we've been monitoring phosphorus for a long time. It's a really important metric. And this is a little bit of good news. As you can see, there's actually a very modest decrease over time in the levels of phosphorus that we're seeing in Candlewood Lake. This is the surface water. Epilimnion is another word for the surface layer of the lake. And you can see it depends on the location. Each of these lines is a different location. It was a little easier to see if I just made them black. But you can see actually they're slowly going down, and these are the measurements that were taken in 2020 and 2019. This is just to illustrate that we're making progress here. This is probably a result of educational efforts that have been made over the past, it's 36 years now that this has been being monitored. But this is not something that's necessarily going to stay this way. We need to continue to work hard for this to continue to go down, or else it will very quickly potentially rise again.
And here we go with saltiness again. And Neil said, Steve, you do the salty. You like the salty. Actually, I've been watching that conductivity, the saltiness in the water for a year now in a very fine-grained kind of way. And it's quite clear what's going on with the trend with that increasing salinity of the lake. And again, it's important to recognize that if you dip your finger in the water and taste it, it's not going to taste salty. We're not talking about that level of salt. But there is a fairly significant increase and a steady increase over the years of that saltiness. And I was just kind of looking over this data tonight, and I was seeing that New Milford, that's the yellow dots on there, New Milford has traditionally been kind of on the high end of conductivity, and the other end of the lake somewhat on the lower end. But in recent years, you'll see that the rest of the lake is kind of catching up with New Milford. If I just saw the first 2/3 of this chart, I go, yeah, I get it, because they pump up from the Housatonic, and that hangs around in the Milford Bay. So we're probably picking up some road salt and stuff like that from the winter when they do the pump up in the spring. But we see now that the whole lake is getting saltier, and the probable source of that would be road salt as development around in the watershed of the lake has increased. We're good, Neil.
Yeah. And the only thing I want to add.
Yeah. I'm sorry. Go back. Yeah.
Yeah. Sorry.
There we go. It's important to talk about the effects of that, and that's what I was leaving out. So that increased salinity has a subtle but continuing effect on the ecosystem of the lake. Certain fish, particularly the fish that fishermen like, are sensitive to increasing specific conductivity. Some of the plankton that provide the diversity in the lake are affected by that. And unfortunately, the blue-greens don't care. The ocean is full of blue-green algae. So they're not affected by the salt. So this salinity is a problem. It's one we may have a real hard time controlling, but it's one to really think about. It's a very clear change that's occurring in the lake, and it's not slowing down at all. Now,
Yeah. Excellent. All right. And so just as a sort of wrap up, saltiness is not included in this, but this is Candlewood Lake's report card for 2020. And actually this is pretty good. So you can see phosphorus here. Our levels are sort of in the middle of mesotrophic and late mesotrophic. So you're talking about the middle part of that timeline, which we sort of knew going in. That's generally Candlewood's identity is sort of mesotrophic. Nitrogen, similarly, right in the middle there, mesotrophic and late mesotrophic. Our chlorophyll A last year was particularly good. It was 2 to 5 micrograms per liter, which is very low, sort of on average. So that's early mesotrophic, even arguably into oligotrophic, that very beginning of that timeline. And then our Secedis transparency, our water clarity, right in the middle here, mesotrophic, but not late mesotrophic. So we're very happy with that. We even got some measurements in this 4 to 6 range, very few, but a few. But on average, we were here in the middle most of the time. So that's why the circle is right there. So that's the report card for 2020. We'll take a look at the one for 2021 soon, but just to give you guys an idea for last year. There we go.
So as I worked alongside Neil and learned from him over the last couple of years, I started to realize that there was a lot we still didn't know about Candlewood. And so one of the things that I undertook myself, if you remember that map he showed a little purple dot at Candlewood Trails. I undertook to sample the lake out in front of Candlewood. That's where I live, Candlewood Trails. I undertook to sample there for to try to do it on a bi-weekly basis to really get a good picture of how the lake changes over time. I did that in the summer when my kids said to me, "What do you want to do for Father's Day?" My son-in-law and I and my daughter were out taking samples. They kind of laughed at that, but that's what we did. As it got colder in early January, I renamed our little runabout the Endurance, as I pretended I was Shackleton, and actually got stuck in the ice one day. And I said, okay, so now I can't sample anymore until, next slide, Neil, until the lake freezes over. And then I got out again and was dropping the probe through there. During that time, when I couldn't really get out on the ice, I didn't have a boat available anymore. If you guys remember, we had in mid-December a big snowstorm. And then on Christmas, Christmas Eve, I guess Christmas and Christmas, the day after Christmas, we had an incredible warm front come through. Lots of rain, temperatures in the 60s, and every bit of snow melted. And that gave me a real good opportunity to see what the immediate effect on salinity was, the conductivity, after a snowstorm. And so I couldn't get out on the water. I had to sample from shore, but I identified a 90-acre kind of sub-watershed right in the community where I live. And there's a stream that drains that whole 90 acres right into the lake. And so I dropped the probe there, and then I dropped the probe near my house and took a look at what was happening with water that was running directly off a place where there had been no road salt applied or anything. And surprisingly, I found that snow melt and incredible rain actually, it actually diluted the water. It was coming in a little less salty, but nonetheless, it was still bringing salt into the water. And so what I really learned from this is that we really have to do more science. We have to continue to do more science. Go ahead, Neil, next one. So another thing that I was able to do, and I'll tell you, I did all this stuff on my own because Neil is a very busy man. And when we go out and do our bi-monthly, it takes for us to do the 4 samples on the lake. It takes us a full day between getting the bottles ready, getting the equipment ready, getting the equipment into the boat, taking the boat out, stopping, dropping the anchor, pulling the anchor up, doing all that stuff on those sites. We have a little odometer on our workboat. We do about 24 miles of lake travel in between all these stops on one of those days. So it's a busy day. It's pretty much a full day of work. And then we have to do Squan's Pond. And you'd think, well, Squan's Pond, it's quick, we get out. But we got to get a boat on a trailer the boat over to Squan's, launch all of our equipment, do the whole thing. So pretty much it's 4 days of the month that we end up just doing this sampling. And the sampling is important, but what I found with my retired old guy time budget, that I could do some more. So when Hurricane Isaiah came through just about a year ago, almost exactly a year ago, when Isaiah came through, I thought, oh, this is a good opportunity. Neil, point to the wind direction on the day before the hurricane, right up, in the first row going across there. Yep. So the day before the hurricane, we had that nice east wind going, gentle breeze. I got up early that morning. The storm was scheduled to rage in the afternoon. So I got up early that morning, went out, grabbed some samples. Those samples show up in white on the column on the chart below. And the storm raged throughout that afternoon. And the next morning, 24 hours after the other one, look at the wind had completely shifted around, died down again. The barometer had risen back up, and I measured the lake again. The lake looked pretty much the same on those 2 days, except for the wind direction. But look at what happened to the oxygen. If you look, we have oxygen numbers in the white column. And then if we compare them to the oxygen numbers the day after, there was a lot less oxygen in the top layers, but look what happened down below. So that crazy wind had mixed the lake to a depth of 25 or 26 feet. And you can see the same effect over in the temperature column. The temperature the day before was 82.5 degrees Fahrenheit. The next morning, 24 hours later, it had dropped 2 degrees, but look at what had happened down deep. That hurricane transferred a huge amount of surface heat down into the bottom of the lake. We really didn't know this stuff before. Another interesting thing way over on the right, if you look at the specific conductivity, all that rain that accompanied the storm actually diluted the water, the salty water, with some fresh water, and the conductivity dropped by a point or two. Next one, Neil. Another really interesting thing happened in December when we started to pump when First Light started to pump up to do a full pond audit in the fall. And if you look, Neil, can you point to Candlewood Trails right there on the profile?
Oh, yeah.
So that's my sample site out in front of my house. So I dropped the probe down there and I got to that thermocline where the water changes temperature. And all of a sudden it was like I saw a different body of water at the bottom of the lake. And that piqued my interest. I was with a friend and I said, boy, I'd like to know what's going on in New Milford. So she said, let's go to New Milford. So we went down to north to New Milford, all the way over on the left there, dropped the probe all the way down. And sure enough, I saw that same different layer of water at approximately the same depth, filling the rest of the New Milford Basin. So then I said, wow, I wonder if that's Housatonic water. And my friend said to me, let's go measure the Housatonic. So we drove down to Boardman Bridge in New Milford, and we were kind of dodging automobiles and snowbanks, and we dropped the probe off Boardman Bridge, and sure enough, we found that the water at Boardman Bridge, just upstream of where they pump it up, where First Light pumps it up into the lake, that water matched exactly that slug of water at the bottom of the Milford Bay and that I had found out in front of my house. So the next day we went out and we traveled south along the lake, taking samples again in New Milford, at the Narrows, at Candlewood Trails, at Oak Island, that's kind of by Gerard's, Chimney Point, and all the way out into Brookfield Bay there by Candlewood Isle. And we saw that that water was indeed, that pumped up water was indeed hugging the bottom of the lake. And pretty much by the time we got to Candlewood Isle, it had dissipated out. There wasn't much of a difference anymore. But this is something we hadn't seen before. And this is something that really, it's important. We know more about it. We know more. Look at the conductivity of the water coming up from the Houstonic. Numbers like 310, 304. See, this is why Neil said, Steve, do the conductivity. Because that blew me away when I saw, whoa, look at the amount of, quote unquote, salt coming into the lake with that pump up. So that was, an interesting experiment. And it wasn't really, it's not particularly good science. I didn't have good controls. This was exploratory science, kind of like cutting edge. Let's just see what's going on. But there it is. This could set the stage for an official study that we could do in the future. Next one, Neil. So we went through the winter, shot down through the ice there. And then First Light started to pump up again in the spring. And if you look at this profile, Neil showed us one, Neil on the left side one, show them depth on the left profile there. So the surface of the lake is at the top of the profile. And there we're measuring the temperature. You can see it was warmest at the surface and then got colder. And then at 4 meters, it turned around and started getting warmer again. Then it stayed the same temperature. Then it got warmer again all the way to the bottom. Very weird. If you look at the conductivity, these are not profiles that we expect. So I collect that data. I come home and graph it and go, okay, that's weird. I wonder if I can explain all of that. And I think I have some explanation to it. And then I did the same thing in front of Candlewood Trails, and I got two profiles that, it's going to take a lot of thinking and more practice to figure out exactly what's going on there. But the pump up into Candlewood Lake is something unique to Candlewood Lake. And so we can't just look at other lakes and say, well, what do they do? Candlewood's doing that or Candlewood's not doing that. And this is all stuff we're learning from this extra sampling that we have going. Next one, Neil, or am I done here? No.
Got one more, yeah.
So I just did this one a couple days ago, and I thought this was pretty interesting. I had a good sample from Candlewood Trails on July 24th of 2020 and another sample on July 23rd of 2021. And I just put the two side by side. I had the numbers from the same depth. I had been able to find the same exact spot in the lake, dropped it to the same depth. And here's an interesting thing over on the left. Look at the temperature this year. We've been running 3.5 degrees Celsius cooler in the top 5 or 6 meters or 4 meters of the lake this summer. That's like 5 or almost 6 degrees Fahrenheit. So it's been a lot cooler this summer. That may have something to do with some of the things that we're seeing. If you look, my old friend, the specific conductivity, we're up from last year on this particular day. And by the way, I compared these dates because this was before Hurricane Isaiah last year. And I thought that the hurricane probably stirred up the lake too much to make a valid comparison. One thing that many of you have noticed this year, we've gotten phone calls about it, is the algae content of the lake. And if you look this year, Neil, go to the Delta BG column there that I kind of drew the red line through. If you look, we're higher pretty much through the top 5 or 6 meters. And, that's the water that we swim in. That's the water that we see. That's the water that we measure the transparency in. That's all there. But if you look down at the bottom, there's actually fewer blue-greens down there. And if you look at the 2 years together, you can see this year the blue-greens are hanging out in the top half of the lake, where last year our big blue-green numbers were in the bottom half of the lake. So we don't know exactly what's going on, but Neil and I were just talking about this the other day, and particularly after I put together the Hurricane Isaiah thing, is that wind apparently makes a big difference in mixing up the water. And we have not, and this is anecdotal, but I know from my going out there last year, I had trouble setting an anchor many, many times. I had water splashing over the bow of my boat while I was trying to sample. This year, it's been calm for most of the year. And we don't see the kind of mixing with the oxygen that we saw last year and stuff like that. So that may be something that's contributing to the higher blue-green numbers that we see this year. And all of that is important to understand so that we can make smart recommendations and plan for how to take care of our lake. Next one. Oh, go ahead.
No, I was just gonna, it's actually a perfect transition. So this is a sort of a good transition into sort of 2021. So one of the other things that, you know, we've noticed this year is we are getting slightly higher phosphorus measurements in the surface, and those blue-green algae really need phosphorus to grow and reproduce and eventually bloom. If they're going to bloom, one of the raw materials they need is phosphorus. And so you can see on the right hand side here on the bottom is that same report card, and these arrows sort of indicate the direction that we're going. This one's a little bit lighter red because if you remember last year, both of these were circled. This year, however, it's been mostly between 25 and 30 micrograms per liter. So it's actually a little higher on average. And we are seeing higher measurements in the surface waters. And so, you know, Steve was talking about how we've been seeing more blue-green algae up in the surface, and part of that likely is mixing. Part of that likely is the availability of phosphorus for those blue-green algae to reproduce and grow and bloom. So sort of evidence that Candlewood Lake is a really complex system with a lot of different things contributing to all of the phenomenon that we're seeing in the water.
It is interesting to note too that we had very little rain last summer, and we've had a lot of rain this spring. And if our phosphorus levels go up as rain goes up, it's a pretty good bet where it's coming from. And that would be from fertilized lawns and stuff like that. So we really have to be careful when we talk about managing the watershed, managing runoff, managing stormwater runoff and things like that, fertilizing our lawns, et cetera.
Yeah. So, and just to go through the rest of this report card. So our nitrogen so far, and I want to, you know, make it clear that this is just through May through July at this point. So this is half of the year compared to any other year that's being compared here. So these numbers can change. They can go either direction. We're talking in averages here with these metrics. So these can still go either direction this year. And what's happening this year doesn't necessarily mean we know what's going to happen next year. But nitrogen so far, relatively the same. So this increase in phosphorus is what we're looking at as a potential indicator of the increase in algal material, which we are also seeing. You can see chlorophyll A increased a little bit. It was here, early mesotrophic last year. This year on the report card, it's mesotrophic, a little worse. And our Secchi disc transparency went down a rung as well. The water is a little less clear, so we went from that mesotrophic level to the late mesotrophic level. You know, we're getting 2 to 3 meters on average, really closer to 2 most of the time this year. So the water is less clear this year than it's last year so far. And just to indicate that again, is that same chart that I showed you earlier, and you can see we've added 2021 here. So just to remind you, this is our algae material here. So on the x-axis over here is lots of algae, and down here is really clear water. Up here is not very clear. So you can see we're here sort of in the middle. We're in good company, lots of years in this area. But 2020 last year, way down here in this clear water area, 2021 is sort of middling. You can see lots of different years in this area, but still generally better than other years we've seen in the past, 2013, 2015, all the way back to 1986. So this is only half a year, like I said, but it's looking to be sort of a middling year as far as our report card is concerned. You're looking at B's instead of A's on a lot of these metrics. I'll hand it over to Steve to give you guys a little update on the zebra mussel situation.
So invasive species continue to be a concern and a problem. So far, we're not infested with zebra mussels, but all of you know that we did find them last year officially, and then the Candlewood Lake community got interested and looked, and we found, what was it, Neil, a total of about 80 last year?
Mm-hmm.
And I won't detail the search of that, but we did a webinar last fall, did we? Whenever it was.
February.
In February last winter, and that's available on YouTube. You can find it at the Candlewood Lake Authority website. But this year, divers did find below the level of the deep drawdown a few more adults. I think 5. Neil, is that right? And but also, and hopefully significantly, no colonies were discovered. These were lone individuals, which we were not sure how they got there. They were adults, though, so they were there last year. And last winter, when we did the deep drawdown to 11 meters, we found some adults there. So even last year, there were year-old zebra mussels at the bottom of the lake. Of course, we've really continued our search. So one of the important things that we did was we are partnered with Westcon to test the water for zebra mussel DNA. And so far, we haven't found any. And that means that the likelihood they don't actively mate. They, spread their eggs into the water, spread the sperm into the water, and they just get lucky. And then they float around until they attach somewhere. So we don't see the levels of sperm and eggs to get a crazy infestation going. Now, that doesn't mean it's not going to happen. We don't know yet, but that's why we're watching very carefully. We have a volunteer network of people who have put together zebra mussel hotels, and those hotels are quite easy to build. You take a cinder block, you tie a rope around it, you throw it off the end of your dock. And once a week, you pull it up to see if there are any zebra mussels there. So far, no one. We had that going all last summer. So far, no one has found any. So we don't know how much the drawdown controls the zebra mussel population, how much the chemistry of the lake controls the zebra mussel population. At this point, we can't make a final conclusion. We can't draw a final conclusion about zebra mussel condition of the lake until we have more information. So we're watching that very, very carefully.
Excellent. And another invasive species that has been here for quite some time now is Eurasian water milfoil. I'm sorry, I'm giving you a little preview of the other slides. So I wanted to give you guys an update on the invasive plant situation in Candlewood as well. Thankfully, so far, there have been no evidence of new invasive plants so far in 2021. The plant survey that First Light conducts every year is being conducted this month. That's a search for new invasive plants as well as monitoring the current status of the milfoil in Candlewood Lake. So They actually give an estimate of the acreage of how much milfoil is in the lake every year. And so we use that information, that's sort of critical information for us, so that we can evaluate our grass carp management program. So for those of you who don't know, in 2015 and 2017, the Candlewood Lake Authority stocked Candlewood Lake and Squance Pond with sterilized grass carp. So they cannot reproduce, but what they do is they eat Eurasian water milfoil selectively. So they really like milfoil. And so what they were added to the lake to actually eat the plants from the top down and bring them from the surface to a lower level where they won't bother you when you're swimming, where they won't get caught in boat prop motors, but where they still exist as an important aspect of the fish community. Fish reproduce in plant beds. So plants are an important part of a lake's ecosystem, but they do get in the way. So the grass carp are sort of a way that we can try to balance those interests for the lake's ecosystem. The last stocking was in 2017. We're working with the Connecticut Department of Energy and Environmental Protection, DEEP, to evaluate the possibility of additional stockings. There's been nothing as far as confirmation yet about stockings one way or the other in the near future, but we are in conversation with them about the possibility based substantially on what these plant surveys tell us. So that's critical knowledge for our grass carp program. We also historically do rake tosses to sample for invasive plants near the beaches, where there's a higher likelihood that you'll see an invasion of a new plant species that may have hitchhiked on a boat or a boat trailer and might enter Candlewood Lake when a boat launches. Thankfully, we have a new program this year that we're very excited about. Those are our lake stewards, and these are employees of that actually are stationed at the 7 public launches around the lake. So you're talking about the 5 town launches and the 2 state launches on weekend days and holidays. And they are offering voluntary inspections for invasive species at the town launches, or at all 7 launches, when boaters visiting to drop their boats in the lake. And so far, from June through the beginning of August, they have conducted 323 boat inspections where they have removed many plants, mostly milfoil, a few other native plants, no invasives of concern yet, but there is evidence that people don't know that they shouldn't be bringing plant material into the lake. And they have educated those boaters on what they should do. Obviously not to travel with plant material hanging off of their boats and trailers when they're switching between lakes. And we are collecting sort of data about where those boaters are coming from. So if they're only boating on Candlewood, or if they most recently visited a lake like Lake George, Lake Little Nona, Lake Sore, somewhere else further away. Just this past weekend, there was a boat from the Gulf of Mexico that went into Candlewood. So this is really interesting information that we're gathering. It's anonymized, so we don't know which boater is matched with, but it allows us to understand where the recreational community is coming from when they visit the lake, and that's really valuable information. But these smiling faces are waiting
up for 1 second
at the ramps to educate boaters. Yeah.
I just wanted to add, so we have found milfoil coming in on a couple of the boats, and it's not just that the milfoil's coming in, but the milfoil, if it's coming from something like Lake Zor or Little Nona, that milfoil could also have zebra mussel larvae on it, or within it,
or adults.
So it's important, even if we say, oh, well, you already got milfoil, what's the difference? It's what could be hitchhiking even on the milfoil. It's very important to have a clean, dry boat and trailer when you enter the lake.
Exactly. Yep. And so these guys are now the first line of defense for Candlewood in protecting us from those. So this has been excellent. We've gotten a lot of really great feedback from voters. They love having them there, and they've learned a lot. So we're really excited about this program. The last thing we want to touch on before we get to questions is blue-green algae. As we mentioned a few times earlier in this presentation, we have gotten additional calls compared to previous years about higher levels of blue-green algae. People are spotting blooms, and we'll show you a few pictures that we've seen that have been reported to us of blue-green algae, more than the past few years. And so we are keeping a close eye on blue-green algae levels. You saw the comparison that we did with our probe and with our sensor that we have. But we also have partnered with Westcon on a blue-green algae weekly testing for the toxin that they actually release. So we talk about blue-green algae a lot, and the reason that we do is because blue-green algae actually has the potential to release a toxin, or I should say a few different toxins, in the water, and that toxin can have potential negative health impacts on people and pets. So this is why we're paying so close attention to blue-green algae. And so we've partnered with Western Connecticut State University as well as with the towns around the lake to monitor the beaches, the town public beaches around the lake every week for microcystin, the toxin that the blue-green algae have the potential to release. And so we post the results from that monitoring on our Facebook every day. This is the post from last week's sampling. And you can see, thankfully, all of the bar graphs. This is actually a bar graph, believe it or not, but the bars are so low that you can barely see them. Thankfully, the blue-green algae toxin levels that we've been getting so far this year have been very low. There haven't been a single measurement that's come even close to this critical point here, 8 parts per billion. In fact, there hasn't been a single sample that's passed 1 part per billion so far this year. So we are seeing higher levels of algae this year in general, but we are not seeing a subsequent increase in the toxin that they release. So this could indicate that while we have higher blue-green algae levels, they are not necessarily actually excreting the toxins. Certainly not anyway in these locations, the public beaches where swimming is even more important than in other parts of the lake.
So if we talk about blue-green algae in the lake, and you should be able to, hopefully you'll be able to identify it. If you recall, Memorial Day weekend this year was a complete washout, rained almost the whole weekend. Right after that, I went out to do some sampling in June, and pretty much the whole lake looked like the picture on the left. Very definitely blue-green algae blooming. Still a little bit, the water was still a little bit cold. I wouldn't have gone swimming probably anyway, but I wouldn't have gone into that water. We don't know if that blue-green algae that we saw that day was dropping off, was excreting toxins. More recently, on July 15th, the neighbor sent me, that's one of the things that once everybody knows you're doing this stuff on the Lake Authority, neighbor sent me this from the swimming area at our beach, and she said, "What's going on here?" And I said, "Blue-green algae, probably stay away." The interesting thing is these blooms that we've seen have not lasted long. This one that you see on the right there she sent me that picture at about 10 in the morning. Blue-greens actually have the ability to come to the surface and photosynthesize, and then they sink back down again. I don't think they're thinking about it. They're single-celled organisms that will cluster in colonies, but these blooms can dissipate within a matter of even a half hour. If you ever get a chance to watch, just sit and watch them. You'll see them coming to the surface, hanging out for a while, and heading back down. So the June 2nd bloom was pretty much lake-wide. This picture was taken in Danbury, but it looked like this in New Milford. The July 15th one was more localized. But I guess our recommendation would be, yeah, hold off a little bit if you see it that thick. But again, even with these blooms, we have not measured high levels of toxins.
Yeah. And if you ever see a localized bloom like this, but the lake looks normal elsewhere, you can switch locations on the lake. This bloom is not something you need to be concerned about necessarily if you go further away and it does not look like this. Blue-green algae blooms, if it's blooming, you'll be able to see it. So it's really a visual inspection for you as somebody who wants to recreate in the lake. Take a visual inspection. If it looks like you don't want to swim in it, we recommend that you don't. We don't necessarily know if it's excreting toxin, but it's a situation of, better safe than sorry. But what you can do is go to a different location on the lake, and that has the potential not to be blooming and to be perfectly fine to swim in. And so just want to wrap things up, just to give a little reminder of the things that we talked about, and then we can take a few questions before we finish up here. But we are very excited that the water quality monitoring program that we've done since 1985 is expanding in exciting and informative new ways. Steve gave you guys a look at some of the really cool exploratory experiments that he's doing. We've been able to double our normal sampling, which gives us a lot more information about what's happening in the lake, which is really useful for our analysis. We want to thank our partnerships with WestCon, with Deep, with the towns, everyone who's sort of made that possible. We're really excited to be working with all these people that are interested in protecting Candlewood Lake. 2020 was a year with clear water and low algae, so it had higher marks on our report card. 2021, on the other hand, lower grades, but middling still. You saw that graph comparing it to other years. Certainly greener, certainly higher blue-green algae content, but in context, middling. So you're talking about bees on its report card. And local blooms, so keep your eyes peeled if you're swimming for those blue-green algae blooms that we spoke about. And we're very excited to be expanding our vigilance both in the lake and at the ramps with our lake stewards for invasive species, including zebra mussels. So if we find out anything more about zebra mussels, if we get a positive result on one of those DNA tests, we will send out that information on social media and on our email list. So make sure to enjoy our social media on Facebook and Instagram mostly, and sign up for our email list on our website at candlewoodlakeauthority.org right here at the bottom. And apart from that, we want to thank everyone for joining us tonight. This was a great presentation, and ask if you have any questions, please type them in the chat box and Mark will read them out to us, and we will do our best to answer them. Thank you.
Thank you.
Thank you, Neil and Steve. We do have a few questions that have come in, so we'll take those now. So one question that came in was asking what the effect of lake activity, such as boating, what effect does this have on the health of the lake?
I could take that one, Steve. So you would think that boating and recreation would have a pretty substantial effect. It sort of seems like there's lots of boats on the lake. These motors are warming up water and putting it back in the lake. So you'd think that, it would increase the temperature at the surface, that you might have oil and other pollutants that are entering the lake. But you would actually be surprised to learn that as far as impacts on the water chemistry, the recreation has actually a very small impact. So there's a lot of water in Candlewood Lake. Just as a way to illustrate that, there is actually when they are pumping up Housatonic water, there is roughly 15 million gallons of water being pumped into Candlewood Lake per hour, and that is still just not even 5% of the volume of the lake. It takes many hours to fill up Candlewood Lake at that rate. So there's a lot of water in Candlewood Lake. So there's not a whole lot of impact that recreation has on the water chemistry. That said, there are impacts that boaters can have certainly on the shoreline. So large wakes can increase erosion, which sort of sloughs off sediment and dirt from the shore into the lake, and that can have a more significant impact than hot water or anything coming actually off of the boat. So we're more concerned about erosion impacts from recreation than we are about the actual recreation itself. The only other thing that
another example of that would be the crowding on the islands that we see on some weekends, and the use that the islands get. Pretty much no more vegetation where people are walking, and then the other things that people do there all add. Again, it's a big lake, but every little bit has an effect. So, and to that end, we're actually looking at starting the rehab of one of the islands, and hopefully, if that is successful, that'll become a thing that we do over the next, I don't know, probably 10 years. Try to get the islands back under control, back to a more natural state.
One other question. I think you sort of maybe touched on this a little bit, but one of the guests noted that milfoil is a big problem, and why is it not a top issue? The cart will work, but why not put more in now?
Yeah, so that's a good question. Obviously, milfoil is a big problem. It's something that we're really focused on with our grass carp program, obviously, and our ongoing monitoring. But we do understand it still has some major recreational impacts, both on the fishing community, on the boating community. Everyone's sort of, when you're in Candlewood Lake, you're going to experience milfoil. And so what we're trying to do is we're trying to balance the ecosystem in Canada. So milfoil is here, and it's not going anywhere. So our ultimate goal now at this point is to make sure that we can try to keep Candlewood's plant and animal ecosystem in healthy balance. And oftentimes, probably still, there's more plant material than is needed. But if we take drastic measures, considering things like chemicals, other things like that, mechanical harvesting, you're talking about potentially having irreversible, strong impacts the other direction, because the plant community actually is critical to the survival of a healthy ecosystem in Candlewood Lake. So it can be difficult to do that balancing act. Our current strategy is the fish and education and monitoring. And we're working with Deep about potentially increasing the number of fish that we have in there. But we're always looking at different alternatives, new cutting edge technologies, new methods that are being used at other lakes that we could potentially adapt for Candlewood. But we are trying to be careful with the ecosystem that we have.
And ultimately, I mean, as you mentioned Deep, I mean, this program that we have is the only way we can do it is through a permit, and of course, so any fish that we put in there require Deep's approval through the permit.
Yeah, that's a good point. We can't just buy fish and put them in. Deep has to, the state has to approve of any additional stockings.
Question about if a boat comes in and has an invasive species on it, or the trailer, whether it's plant or animal. Wondering what the process is, if they are turned away, or if the what the process is for that protection. Sure.
Steve, you can jump in if I missed something, but basically if it's a plant, well, I should start with, our goal is to have every boat be cleaned, drained, and dried before it enters the lake. And so that's the best way to ensure that they're not going to bring in any invasive species, plant or otherwise. But if our stewards, for instance, were at a ramp and they were visited by a boater with plant material on their trailer or on their boat, first and foremost, they would remove it. Thankfully, with plants, you actually generally need at least a piece of the physical visible plant material for it to be able to reproduce. So if you remove all of the plant material, it's a little easier to manage than something like a zebra mussel, where if you have a boat that tries to enter the lake that's covered in zebra mussels, you are really required to take that, wash it with hot soapy water, dry it completely, and then it's been fully made clean from zebra mussels, and to drain anything like the bilge, live wells, any potential ballast, any water from potential other bodies of water. And so, that has to generally be enforced by legislation. And in the state, the current status of invasive species legislation doesn't actually allow us or our stewards to enforce inspections. But most people generally don't actually know these rules. Many don't even know that, these plants or animals hanging off of their boats are an issue. And so they're happy to talk to our stewards. Our stewards can educate them, and they're happy to do the right thing because they love Candlewood Lake just like everyone else, and they don't want it to get hurt certainly by their impact. And so we found that a lot of boaters have been willing to learn from our stewards. And if thankfully they haven't seen a boat covered in zebra mussels yet, but if they do, they'll ask them to go to a car wash that can support cleaning that boat in such a way that they can enter the lake safely.
Are there any other questions related to water quality, environmental review, and speed? All right, I think that's it. We are in a few minutes over an hour, but not too bad. So
excellent. Yeah, well, this was great. Thank you everyone for joining us. We really enjoyed it, and stay tuned on our website and on our Facebook and email for future webinars. We'll plan soon, hopefully.
Good night, everyone.
Good night, guys. Thank you.